This lecture is about growing up and growing old. And if Frankenstein has made his angel, then he'd like the angel to live as long as possible, remain as young as possible, and if possible, if it lost a wing or an arm, to be able to grow a new one. Let's start with an experiment. Let me introduce you to John Wade, who's from the inner magic circle, and I'm now going to turn things over to him. John.
Thank you very much, Lewis. Well, now let's, good afternoon to you. Let's just pick out a volunteer who'd like to help me in an experiment. Oh, look at this. All these hands go up, and you don't even know what you're volunteering for. I'm a member of the magic circle. There's a lad there. Would you like to help me from just down there? Give him a little encouragement. He needs everybody's look. That's good. My name's John. What's yours?
Robert.
Robert. Yeah. Okay, Robert. Are you at all familiar, Robert, with one of these things?
No, it's one of my things.
You have one like it. Has it got a blade that long?
No.
No, it doesn't have. It's quite a fierce blade. Would you like to? It's not plugged in. You're quite safe. And it doesn't do anything nasty, does it? It doesn't fold up.
No.
It really is made of tin, rather nasty. Okay. Well, I'm going to ask if somebody would just, Bryson, if you just plug that in for a minute. We'll just take this for a second. Would you hold one end of that for me? That's a nice loaf of crusty bread. And we'll just see if the thing's working. Hold on. What a lot of crumbs. You realise in a few minutes it might not be crumbs. Are you nervous?
I think so,
Yeah. That's two of us. Right. Would you like to kneel down there? It's quite a suitable pose. Now, don't worry, I've done this with several people in the past and only lost two of them. So, now if you just put your head down there a little. That's the lad. We've got a little device here that comes in and sort of locks your head in from the top, like that. And I've got a little bit of toweling here for reasons, and it's the right sort of colour, but we won't go into that. Right. Also, there's a little hole in the top. If you just nod your head there, there's a little hole in the top through which I'm going to put a carrot, and I think we can just see that in the top there. Okay, then. Now, wait a minute. We've got a little thing here. There's a basket here. And if you put your hands one each side, you might just like that. You might be very quick and catch whatever goes past as you go. Right, well, this is the big moment then. We put the thing in like this. Are you quite comfortable? Right. There goes the carrot. Don't nod. Are you all right?
Yeah.
You really are? Get up. You've done very well indeed. Absolutely splendid. Don't go away. Don't go away. We've got a little certificate for you which qualifies you as a fully paid up member of the 50-50 club. Just don't nod for a little while. There's no incision there. Thank you very much indeed for playing the game. Thanks a lot.
John, thank you very much for doing that very interesting experiment for us.
Anytime with anybody you like.
Thank you very much indeed. Can I offer you a note of caution, please? Do not try and do this experiment at home or anywhere else. It's a speciality of Mr. Wade's, so please, please don't try and do it anywhere. Well, you can see it is possible to cut someone in half and join them up together again. And there are animals with which that's possible. And one of those animals is a small animal that lives in ponds, and that's Hydra. Do we have a Hydra? Here we have it. Lovely. We have a beautiful Hydra here. And you can see it's stuck down on the dish there. And there's some tentacles growing out the other end. And these are buds coming off. Now, it's possible, and Amata is going to do that, to cut the animal in half, and then we'll see what happens. Amata, could you cut that in half for us, please? One quick nick, and it's in half. Now, the Hydra recover from that perfectly happily, and they regenerate at the one end. The one with the tentacles regenerates a sticky bit that sticks to the dish, and the other end that's sticking to the dish will regenerate tentacles. And we've got some, it takes a couple of days that, and we've got a couple here that show that quite nicely. Could you just point out? Oh, here's the end, which is actually beginning, which is the end? Yes, that's the end there, which is beginning to regenerate. It's healed over. And then a little while later, this is after a couple of days, it begins to form, and you can see the small tentacles there. And then after 2 days, it's almost back to a normal animal. And in fact, Hydra reproduce by splitting, and so this is not a traumatic process for them at all, and quite a normal one. Now, there are other animals that regenerate, but they regenerate in a different way. The way Hydra regenerates is not by growing. It remolds the tissue. When Amata cut the animal in half, each piece didn't grow initially, but simply remolded the tissue there so as to form tentacles on one end and a sticky foot at the other. And the way we know that it's nothing to do with growth or cell multiplication is that we can block cell multiplication by giving the Hydra an enormous dose of radiation. We can give them 20,000 rads. Now, if I give you 500 rads, that would be quite serious. So one can give the Hydra an enormous dose of radiation. Not a single cell will divide. The Hydra will drop dead at the end of 3 days, but they'll still regenerate. And that shows it's remolding and it's nothing to do with growth. However, other animals, like newts, can regenerate their limbs if they lose them. And that certainly does involve growth. And let's have a look here. This is the limb of a newt. And if the newt loses its limb, say at this level here, at the level of the humerus, then it heals up and a new limb begins to grow out. And here you can see it getting longer and longer, and even a little hand plate there now. And then eventually, after a few weeks, and this is not a single animal, this is different stages in the regeneration, you'll see it's back to an almost normal limb. Okay. So newts can grow new limbs when they lose them. You may think that what's happening there is the cells in the limb are saying, well, we've lost part of it, let's try and restore what we originally had before. And the question is that true? And we're going to investigate that with an experiment on cockroaches, which often lose their limbs and can regenerate them too. Let's see if we can see the leg of the cockroach. Some of these have lost their legs. Let's see if I can find a nice leg. There's quite a nice leg there, and the particular part I want you to look at, oh, there's a lovely one. You see it with the spines there? That's the tibia. That corresponds to that part of your leg, and that's the bit we're interested in. Very nice one there. There it is again, the spiky bit. Because what we're going to ask about that leg is whether the cells really know what they're doing. And in order to do this, I want 2 volunteers, please. Let's see. Let's take you from right up. Yes, you. Thank you. And let's see if we can find someone from there. Yes, we'll take you. Thank you. You can come down. That's lovely. Thank you. Sorry, what's your name? Yvette. Yvette, will you come and stand this side? And what's your name?
Thomas.
Thomas, if you stand that side, I'll show you what we're going to do. It's not going to be with cockroaches, just live ones anyhow. We've got some models here, and I'll show you what it's about. These are models of the cockroach leg, or the tibia of the cockroach leg. Thanks very much. Okay. What I want you to think of is this is that tibia of the cockroach. And the regions are numbered. The cockroach, think of that end, and the claw, the foot down that end there. So this is just the tibia, and it's not segmented. It's just that we've numbered these regions because the cells, we think, have positions down the leg. And the first experiment we're going to do is we're going to cut it here, in these regions here, at level between 9 and 10 and between 3 and 4, take out this piece and join these two together. So if you just cut it effectively by untaping that there. Terrific. Would you keep that piece, because that's going to be your piece, and that's your piece. Okay, so we've cut that out. Could you join them together, please? That's fine. So if you just stand to a side, Thomas, because just otherwise others can't see it. And hold it. I want you to hold it, please. I want you to hold it too. That's fine. And so we've made it much shorter. And so we've joined region 3 to region 10. And the question is, what's the cockroach going to do? And the cockroach realizes that something is wrong. And if you just hold on to that quite tight and just separate it. You pull too. Slowly, slowly, slowly. So we're getting 5, 6, 7 appearing. 8, a little more. Stop there and hold. 9, a bit further your side. Off you go. Terrific. Just stop there. So what's happened? It actually regenerates segments 4, 5, 6, 7, 8, and 9. And so the leg's back to normal. Very clever. And so what you may think is happening is the cockroach says, oh, I know I've lost the middle part of my leg, and I'm going to restore it. Let's find out if that's really true, because we'll do another experiment. Okay? This time, we're going to take the leg and we're going to join similar regions together, but we're going to make the leg longer. Would you cut it there just the way you did before, please? Terrific. I'll take that away. Get rid of that too. Would you take this leg here, cut it there. Thank you very much. We'll take that away. And if you could hold that one, and you hold that one, and we join them together like that. So hold on, just keep them like that. So what we've joined now is the level 9 to the level 4. Now what's the cockroach going to do? Well, you may think, and if you push it in, it's going to get shorter. Push it all the way in, as far as it will go. Terrific, let's put that in the center. But that isn't what happens. It doesn't get shorter. The cells don't do that. If you pull it out, let's see what happens. Slowly. Slowly. Keep pulling. You pull that way. Slowly. There we are. Look what's happened. It's got even longer. Because what we see here that has happened is that the missing parts in between the bits missing between 9 and 4 have actually been made. And we can see this on the slide here. There's the normal leg, and here's the one that is made longer. And so what is quite clear here is that the cockroach doesn't know at all what's really happening. All the cells are doing are following a local rule, that when there's a difference between the 9 and the 4 or the 3 and the 10, just the missing cell positions are filled in. So cells perhaps aren't as clever as they might seem at first sight. Thank you very much for your help indeed. Thank you. Now, in that experiment, the way we did things was to look at position along the leg, and you could think that the cells were responding by filling in the missing positions. And you could think that the cells knew their position, perhaps because there was some chemical concentration gradient along the leg. But now what I want to look at is what happens when you switch things, when you switch a piece from one leg to another, because we're going to have to look not only what happens along the leg, but since the cockroach leg is a tube, what goes on in this direction. Here are 2 cockroach legs. Just the same we've had before, but this time we're just looking at one portion of them. And what I want you to think of them as, I want you to think of me as the cockroach, and here I lie like this with my 2 legs. So here are 2 portions of the tibia. And just let me show you that what we're particularly interested in this time are the numbers around this part of the leg. It's almost like a clock. Here it goes 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay. And now we do the experiment. What we do is we cut here and we cut here. I cut here, that's right, I take that away and put that there. And I cut here and I take this away. This one's going to regenerate perfectly happily. But what I'm going to do is I'm going to take this piece and I'm going to put it on to this one here. And the first thing that you see when I do this is that, in fact, there's a lack of correspondence between the numbers. There's no way I can move a piece of tubing from this side to this side and still keep the numbers matched. There has to be there must be a discrepancy in the numbers, and I've just arranged it so I've put the 6 next to the 12 on top here. First of all, let me tell you the result of an experiment like this. You just put it more in the center. The result of an experiment like that is, there's the normal cockroach tibia, and in that part where the cut is made, you get 2 extra limbs growing out at right angles. And we want to try and understand that. So at this cut surface, you're going to get 2 new limbs growing out, and we want to try and understand it. We already have one rule. What's our rule is that when 2 regions are next to each other and that they don't have the same number, then you fill in the missing numbers. But I've also got to tell you an additional rule, that in order to get regeneration, or whenever you require a full circle, you need all the numbers round the clock. And wherever you get all the numbers round the clock, you're going to get regeneration of a leg. So let's just actually see what happens when we do this. I'm going to move them apart a little so as I can actually fill in the missing numbers. I want to screw that down. Okay. Let's have a look here. 3 corresponds with 3, so nothing goes in there, nothing of interest. 4 is opposite the 2, so I've actually got to fit in a 3. Is that correct? It is. Good. Here I've got a 1 and a 5. I've got to put in a 2, 3, and a 4. There, I've done it. And here I've got a 6 to a 12, so I've got to put in a 1, 2, 3, 4, and 5. And let me do that. So I'm just following our old rule exactly the same way, but in a slightly different way. Let me do this on the other side now. Between the 8 and the 10, I put in a 9. Between the 7 and the 11, I put in 8, 9, and 10. I'm bound to knock the whole thing over. And then the last one, of course, will be 6, 7, 8, 9, 10, 11. And what do I have here at the top? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. A complete circle. What's the rule? When you have a complete circle, you regenerate a leg. And that's why, on both sides there, you get these extra legs going out. At least that's one of the best explanations we have for this very remarkable result. And that's understood in terms of the cells having positions. They know their position. Okay. What about the newt now? Let me show you another experiment with the newt. And we wish we understood how newts did regenerate their legs. And I'm going to do, you've got to think of me not as a cockroach this time, but as a newt. And now we do the following experiment, which was done many years ago, and I'm going to repeat it for you. Excuse me, I'm going to undress. I'm going to make a little slit in the belly of the newt. And I'm going to put the hand in there to get a blood supply like that. So there's now a blood supply going to this arm. And so now what happens if a cut is made here? There'll be 2 surfaces from which you can get regeneration. Okay, we know this one is going to regenerate an arm. But what's this one going to regenerate now? Will it regenerate me? Let's have a look. here's the experiment. There's the newt's arm that we're interested in. Then we tuck it into the belly like that, make a cut there. Well, that's how the experiment was done. Then we've got two surfaces from which we can get regeneration, this one and this one. And the question is, we know this one's just going to give a normal limb, but will this regenerate the newt? Let's have a look. No, it does exactly the same. Those two cut surfaces simply regenerate arms. And you can see once again, just as in the cockroach, the cells aren't being clever in trying to reconstruct the arm, because there is already a radius and ulna here, and they make another one there. So it's just a local rule that they're following in relation to regeneration. We'd love to know why we can't regenerate our arms, although I should point out to you, you can regenerate your liver. And if when you're very young, you lose the tip of your finger, so long as you've still got your nail there and the wound is kept open, that will regenerate. Lots of people work on how newts regenerate their limbs, because it really would be very important to understand. And one recent very interesting result is that you can actually trick the cells into thinking that they're in the wrong position. Normally, if you cut the newts, or if the newt loses its arm there, it will actually regenerate a hand. If it's cut there, it will regenerate the whole limb. But if you cut here, and the animal is placed in a solution of vitamin A, these cells think they're in the wrong place. They think they're up there, and so what happens, it begins right from the beginning again. And instead of making a hand, it begins to make a humerus, a radius and ulna, and the rest of the arm. And so here is one such example. Humerus, radius, and ulna, and normally it would have regenerated a hand, but the vitamin A has told these cells that made these cells think they're in a position up here, and it starts again and makes a humerus, a radius, and ulna, and a hand. And this may give us some clue as to what's involved in regeneration. What I want to talk about, though, a little bit more now is growth in the more general sense. There's no question the regeneration of limbs involves growth. But what I'm more concerned with now is our overall growth, and you all know that you grow a great deal from the time that you were a baby. And in fact, if you go back to what's happening in the womb, by the time you're about 6 weeks to 2 months old in the womb, you really do look already like a little human being. And over the next few months, up to the stage of about 4 months, a great deal is simply involved in simply getting bigger. In other words, the main structures are laid down, and it's a question of growing. And it's that growing process that I really want to concentrate on. And one of the things that you have to think about when you're growing is, how is everything organized so that it's coordinated? How is it that your skull, for example, just fits your brain? In other words, if you have a very small brain, why doesn't it rattle about inside your skull? And the reason is that the way the skull grows is like this. This is a model of a 7-month-old baby. And you can see here the bones of the skull are separated by these lines here. And there's this open region here, which in fact remains open when you're a baby till you're about 18 months old. Bone is laid down at these joins, and it's due to the growth of the brain that stretches these apart and causes the bone to be laid down. And in that way, the growth of the skull exactly keeps pace with the growth of the brain. So the two are coordinated in a very nice sort of way. And that's why your skull fits your brain in a very sensible manner. Let's take another example, and let's have a look at the limb bud. And here is the limb bud on about a 4 to 6 week old embryo, perhaps about 4 to 5 weeks. And here's the limb bud. And here's the human embryo. There's the eye, and this is the brain over here. Now, already at that early stage, certainly your humerus, this region here, is already laid down. And it's laid down very small. And yet you see it gets very big. And the problem is, how does it do it? And is how are the muscles and the tendons all organized so that they all come together in the right sort of way. So your humerus is laid down where it's very small, it grows a great deal, and the reason why your muscles and your tendons are just the right length in relation to it are due to the fact that as it grows from that very early size, the tendons which are attached to the bone, and which are sort of like strings, and which are attached between the bone and the muscles, as the bone grows, the tendons pull on the muscles, and it's that pull on the muscles that makes the muscles grow. If there's no pull on the muscles won't grow. And it's because they're pulled that they end up at the right length. The next question is, why are your limbs, well, they should be about the same length, and indeed mine more or less are. That's a problem, because I told you that your humerus and your limb is laid down when you're very small. It's going to grow for about the next 18 years, and they end up about the same length. Is there some clever way in which the left arm knows what the right arm is doing? Do they send little messages to each other? And the answer is they don't. There are no messages between them whatsoever. They grow completely autonomously, and it's rather like letting off two spaceships about 18 years ago. They go their own way, and in spite of all the interplanetary dust and all the knocking about they might get, after 18 years, they get to about the same position. And I think that's a rather remarkable feat, and it's rather clever of the cells that they can actually do so. And you can see this very clearly, this autonomy, or the fact that the limbs grow quite independently, if we do the following experiment. We go along to an amphibian, an axolotl, and there's a large species and there's a small species. And we can graft the early limb bud from the large species onto the small species. Now, if there was some communication between the limbs, you might think that they'd try and even up, but actually have a look what happens. There's the bud from the large axolotl that was grafted on, and you see it just does its own thing and grows a very large limb, and this one simply remains small, and these two are a perfectly normal size. There are abnormalities in people, they're rare, in which the limbs aren't the same size. They do grow to different lengths. And here's an illustration of one. It's one of the earliest illustrations in the medical literature, about 120 years ago, in which you can see that one leg is enormously larger than the other. And the reason for that is largely due to the fact that there's too much blood supply to this leg compared to that one there, and that's why that one has grown to that enormous size. But on the whole, the two sides of one's body, with the small differences, really keep very well together and actually grow to the same lengths. Having said that your humerus and your bones grow, let's look at your overall height and try and have a look a little bit more clearly how these individual elements actually grow. Now, what I have here is a normal growth curve. Here's the age, your age, average age, well, no, age along the bottom, and height in centimeters here. And something like 94% of people will fit within these bands here for the normal growth curve. So you start off small, and you actually get taller. And notice here, there's the adolescent spurt. You know, you suddenly at adolescence go through this rapid growth phase, and then, of course, your growth stops. Now, what's actually happening here while you're growing? And let me try and explain to you. A great deal of your growth is in your long bones, like your arms and in your legs, your humerus and your femur and so forth. And the way these grow really involve a rather special mechanism, and the special mechanism is the growth plate. And that's a thin cartilaginous region in the bone which actually gives the growth. Bone itself does not grow in your long bones. The way you get taller is due to the growth plate. And let me show you a growth plate. Here are some X-rays of an arm, and at the edge near the joint in each one, there's this dark region, these dark regions here. And that dark region there is dark because it's cartilage and therefore doesn't block the X-rays. And that's the growth plate. And that's the region where the cartilage cells are growing, very thin, just a few millimeters thick. The cartilage cells grow, and then they are replaced by bone. So all your growth, particularly of your long bones, is simply due to the cartilage growing and then being replaced by bone. Okay? And we can see that there's a growth plate there, another one there. In fact, most of your bones have two, one at each end. And if you damage that growth plate, for example, playing sport or something like that, there'll be a reduction of growth in that particular region. Now, here are two hands of two children of exactly the same age. As you probably know amongst your friends, some of you suddenly get taller. Friends who have been quite similar height, suddenly one gets more tall than the other. In other words, your growth spurt comes at different ages. And also, you stop growing at different ages, and you can see this very clearly here. In this hand here, there are lots of growth plates in the digits. There's nice growth plate there, nice growth plate there. All these black lines here are growth plates. But exactly the same age child, have a look at this one down below, there are no growth plates at all. And the reason there are no growth plates is that the growth plates have disappeared, and if there are no growth plates, there is no growth. So this hand, or these hands, have stopped growing. This one's continuing to grow. So the presence of growth plates is essential for growth, and the reason why you stop growing is normally due to the fact that your growth plates actually fuse up and disappear. And to illustrate the point about growth plates, just let me tell you a story here about a child who ate some paint when he was young and therefore ate some lead. And what lead does, it slightly interfered with the growth and left a little mark at the time of the growth. And I'll just show you here. It leaves a little line. There's one there, there's one there, there's one there, and there's one there. But let's focus on this one here. Here's this sort of slightly light line here. Reflects the fact that when the growth plate was in that region, the child ate some lead, and that sort of led to a little disturbance there, which made it slightly more dense, and it's rather like a tree ring. And so, in fact, growth plates can be thought of rather like the way a tree grows, and in this particular case, it actually left a ring behind. Now, because of the importance of growth plates in our growth, we can now begin to think of what can go wrong with growth. And very, very helpfully, we've got some people here who are going to illustrate this. And very kindly, she's come along. Sandy Pym has come along to join us today in order to talk about, she has restricted growth. And Sandy, when was it first known that you were going to be shorter?
My parents first realised when I was 2, but I didn't realise really until I was 10 that I was shorter than other people.
Are there other short people in your family?
No, there's no one else in my family who's small.
Am I right in thinking, Sandy, that the reason why, I mean, your head is perfectly normal, yeah. But that your limbs are short is due to the fact that your growth plates simply haven't grown?
Yes, and it was also due to a spontaneous genetic mutation.
So it's just a spontaneous mutation which has just unfortunately occurred in you.
Yes. And
that your growth plates...
of course, it didn't grow as well.
Was there any suggestion that anyone could do anything to help this in any way?
No, because they just stopped growing, and so I ended up like I have.
Sure. I think it's important, in Sandy's case, it's just that the growth plates, as I understand it, that the growth plates don't grow, and that it's not really fully understand why that happens. Sandy, may I ask you your height?
I'm 4'4".
And that's how many centimetres? About 130 too.
Yes.
Thank you very much indeed. I'm very grateful for you for coming along. Sandy, I'm going to put I think it's about 132. I'm going to put you there. You'll see, you're really, as you know, you're really quite a long way below the normal level. Now, that's one way in which growth can go wrong, in which the growth plates, for reasons which we simply don't understand, just don't work properly. But there are other reasons why people can have restricted growth. And let me talk to someone else who has that. Because another person who has restricted growth is Martin. And Martin, as I understand it, you were a bit shorter than normal a little while ago. Is that true?
Yes.
How old are you now?
8.
But you've been growing a lot recently.
Yes.
And why is that?
Because I've had this growth hormone and it makes you grow more.
Has it made you grow a lot more?
Yes.
Have you noticed yourself shooting up?
Yeah.
How often do you take the growth hormone?
Every evening.
And how do you take it?
By injection.
And who gives that to you?
Mum or Dad.
Your mother gives you the injection every day.
Yes.
And have you noticed that your height has increased significantly now?
Yes.
And does that please you, I hope?
Yes.
Terrific. That's very nice of you. And I'll show you. I'm going to put your height on here. And as Martin made clear, when he started taking growth hormone, that was about what?
A year ago.
About a year ago. Okay. So I'll put that on here. So you're now 8, so it's about when you were about 7 there. That was your, let me stand this side, I'm doing it badly. On this side here, that was your growth before, so you were really quite a little bit below the normal. Then you started taking growth hormone here, and for about a year, and you've actually shot up, and now you're within the normal range. That's very impressive. I think it's very nice. And incidentally, I think it's very impressive that growth hormone can do that, and what Martin's problem is in fact that he doesn't have enough growth hormone. Nothing whatsoever wrong with his growth plates, and Martin, as I understand it, you're going to really have to take it probably every day till you're 18.
Yes.
A lot of growth hormone you're going to take. Now, and also, I should tell you that growth hormone comes from the pituitary gland. It's actually made in the pituitary gland, which is in the middle of your head. Okay. Now, if Martin doesn't grow sufficiently because he hasn't got enough growth hormone, what one has to think about are other cases. And Christopher Greener has very kindly agreed to join us. Christopher, how very nice of you to join us. Welcome. Christopher is you are the tallest man in Great Britain.
Tallest man in Britain, that's right.
How tall?
7'6" and 1/4 inches.
Yes. Tallest man in the world?
Tallest man in the world is, I think, 8'2".
I mean, he's going to be even, yes. I could well imagine. Do you come from a tall family?
Reasonably tall, yes. My father was 6'3" and Mum was 5'6". So they weren't short, but they weren't exceptionally tall.
And when did you realise that you were going to be taller than most?
It's very difficult now to look back and discover the actual sort of sizes I was at various ages. But when I left primary school at the age of, 10 or 11, I was as tall as, if not slightly taller than most of the teachers.
Well, an advantage for some. Christopher, am I right in thinking that it's because you do have too much growth, or you had too much growth hormone?
That's correct. Basically, in very, very basic terms, I had an overactive pituitary gland, which produced too many growth hormones. In fact, I think I had about over 20 times the normal amount of growth hormone output.
And so that's the result of...
That's right.
Christopher, I'm enormously grateful of you to coming along. Thank you very much indeed. Thank you. Please come and sit down here. Thank you. And I should put Christopher up here. And of course, that's Martin there. Growth hormones made by the pituitary, as Christopher told you, but there are other hormones that also affect growth. And one of the most important of these, particularly in making the difference between men and women, is the hormone testosterone. If you're female, it's different, but if you're male, males, because they have a Y chromosome and they develop a testis, actually make a large amount of testosterone, and that testosterone has very important effects on the growth of males, and in fact the external appearance, the difference between males and females, is largely due to the fact that the female embryo doesn't have testosterone, whereas the male does. Now, if you look at the genitals, it's very interesting, because at 6 weeks of age, it's quite impossible to tell in the embryo any difference between the genitals of a male and a female. They start off looking exactly the same. So both male and female at about 6 weeks, 7 weeks look exactly the same, but this one's developing into a female, and that structure then develops into the clitoris and the typical female sexual organs. However, in the male, exactly the same structure, under the influence of testosterone, this time develops not into a clitoris, but into a penis. And so you can see here quite clearly that it's not the difference in external appearance between men and women isn't in an intrinsic difference in the cells, but rather due to the effect of the hormone testosterone. So we've seen how hormones can have a profound effect upon growth. What about immortality? Could we live forever? Let me tell you a little story. One of the daughters of the gods fell madly in love with a man called Tithonus. And she went along to Zeus and said, "Please make Tithonus immortal." Zeus did so, and Tithonus was made immortal. But he became very unhappy because she had forgotten to ask also that he should not age. With the result is he was immortal, but got older and older and older, iller and iller, and finally pleaded to the gods to release him and let him die, which they did. And so we have the problem of aging. And that's really quite a difficult biological problem. We start off fresh-faced and young, but with time we grow, and one doesn't get the sense of aging at this stage at all. In fact, we're continuing to grow, and it's hard to believe that we are aging at this stage. However, there does come an age at which we do believe that we begin to age, and perhaps the best way to think about it is in terms of wear and tear, although it's really quite difficult to understand exactly what's happening. If only we could understand what is happening during aging, we may be able to do something to prevent it. Now, I think wear and tear is a good way of thinking about it. Like that. And I'm going to ask me, what I skin, take a nice big lump, hold it up, that's it, hold it up, quite tighter than that, and let it go. You see it goes back, I regret to say, rather slowly. May I do it on you? Almost instantly. I'm afraid that you can try it on your neighbors if you like. It's a well-known way of telling how old someone is, because I regret as you age, the skin goes back more and more slowly. It's wear and tear. And aging is a deep problem. Just let me give you a feeling of why I feel it's such a deep problem. Consider two animals with the same sort of basic tissues, a mouse and an elephant. An elephant is only born after 2 years, or slightly less than 2 years gestation. And you wouldn't call a young elephant old. However, a 2-year-old mouse is very old. So here we have the same sorts of tissues, the same sorts of cells, in the one case in the elephant newborn, in the other case in the mouse very old. We need to understand what that means, and we don't understand it. One way of thinking about aging and longevity is to consider how we fare with regard to death now as compared to, say, about 100 years ago. And let's think of 50 of you in the seats here, and what the situation would have been 100 years ago. Of the 50 of you in the stands now, 1/4 of you would not have been here because you would have died by the age of 4. And by the time you were 20, another 5 of you would have to go. However, nowadays that's not the case, and now you're all here because only 2% of you would not have reached the age of 20, I'm glad to say. Okay, let's have a look at that in terms of what it looks like, in terms of survival. And here what I'm going to show you are some survival curves starting off with, or thinking of 100% at the start, or 100 people, and age along here. And this is what it looked like in the Stone Age. Lots of deaths very early on, very few people reaching an old age. About 100 years ago, here we are, not many people reaching an old age, and not many people getting very old. And now we can see something very different. The survival curve right up here, and what you can see very clearly now, that when you die, very much depends upon how old you are. Quite a different situation. So first of all, we can see that aging really affects your chances of survival now. And the other thing you can see is that even from the Stone Age, there's not much change down this end. It's as if there's a wall here beyond which really almost all of us cannot pass. And we have no idea what that wall is or how to alter it in order to get, say, to 150 years old, 120 years old, or 200 years old. A lot of people about aging and what and how it actually takes place. It's a very difficult problem, as I've already indicated. One class of experiments that people do, which may give some indication or an experimental system as to what's going on during aging, involves the following experiment. You take a dish, and you take some of the cells from my skin, you put them into this dish, let them grow. They grow until they cover the bottom of the dish, and then they stop growing, and they won't grow anymore. However, if I take those cells out of there, divide them into two, and put them into two new dishes, they'll start growing again until they cover the bottom of the dish. And then when they do that, I can subdivide them again, so that's yet another doubling. I'll do these ones here too. Then I can take this dish, and when it's stopped growing, I can divide it into two, and this one into two, and this one into two, and the last one into two. And the question is, could one go on doing that forever? Will my cells just go on growing and growing and doubling and doubling? And the answer is they won't. That in my case, after about 20 doublings, the cells will just stop. No more doubling. They won't die, they just won't go on. If I took the cells from a sort of 12-year-old in the audience, they would double many more times. They'd go through about 40 doublings. A newborn baby, about 50 doublings. So there's a finite limit on the number of times the cells will double in culture. And this has been thought of as possibly giving a clue as to how we actually age. Doesn't really solve the problem of aging, but this class of technique of making cultures of cells can be used in a very positive way, as let me show you here. What one can do is one can grow skin cells in culture, and what Amatra has done here is add a little enzyme so that the sheet of cells at the bottom of the dish have actually come off, and there you can see it as a sheet. And this is used clinically now, because what you can do is you can take these cells, you can put this gauze on the cells like that. You can see what you can do then is you can take those cells and apply them to someone's leg with a skin ulcer. And so this is the top size of the gauze. The cells are on the other side. This provides a very powerful technique for treating skin ulcers and, for example, even treating burns, because the cells now bring about a remarkable improvement in that condition. In these lectures, I've been trying to see how Frankenstein could construct an angel. That is, by not going to the slaughterhouse and the dissecting room, but really by seeing how we alter development, because as you'll know, we all come from a single cell, the egg. And as I think you'll see, he really has a very long way to go. It's really a very difficult problem to know how to construct an angel or even a human being. What I think he ought to have learnt if he's been listening attentively is that the real problem that he's faced with is how the genes control development. Because you have in the nuclei of the cells this genetic information, and that somehow controls what the cells do. And that's a real problem, how one goes from genes with their messages to the 5 fingers of the hand. It's a really very difficult problem. And the reason why it's so difficult is that genes, the genetic information, they just contain the messages for proteins, okay? They wait there rather passively. They then give the message for a protein. A protein is made, and that sets in motion a whole cascade of events, not only within the cell, but between the cells. And it's because there are so many interactions, it's very hard to predict for the brain, for the arm, for the eye, how a change in a gene or a change in a message is actually going to affect what the structure or the structure that we're actually going to see. It's extremely difficult to predict. Even if we knew everything there was to know about the messages in the genes, it really at this stage wouldn't enable us, or wouldn't enable Frankenstein to construct the angel, because we simply don't know the sequence of events that lead to the constructions of arms in terms of what the genes do and how they make the proteins. However, I don't want to paint too black a picture. Developmental biology is really still a young subject, and we're only at the beginning. And there are some general rules. For example, we saw that using the sort of rules of origami, that one can construct quite complex patterns with very simple rules. In other words, what the egg contains is not a description of the adult, but a program to make it, and that can be really quite a simple set of rules. Another general principle, which I hope he will have learned, that position is terribly important in the life of a cell. A great deal of what cells do during development depends upon their position, and it's a major problem to try and understand how they find out what that position is and also how that turns on particular genes. I think there's quite a lot of anxiety about genetic engineering, about cloning. There is the idea that perhaps, well, biologists will try and make lots of identical children. I can't possibly see why they should want to do that, or who would bear them, or what would be the purpose of it. And just let me tell you, even if it were the case that it were possible to make children like that with the identical genetic constitution, and that is, the brain would be wired up more or less the same because there is variability even in identical twins, the experience that one has in life can have actually a profound effect on how we actually behave. It's not just development, although there is a profound biological inheritance. If we look to the future, the place that I would look for progress is the fly. And the reason I look to the fly is that's where genetics and development come together. We know a great deal about the genetics of the fly. We know a great deal about its development. And there's a real chance of finding out how things work. And what's particularly exciting is that some of the major controlling genes in the fly are also present in our own cells. I'm always very nervous about making predictions about the future. We enter the future backwards, as Jacques Prévert said. You should be very suspicious of any scientist who tells you what sort of discoveries and what possibilities there are. It may well be that it will be possible to grow cells in culture so that when someone has a heart attack, you can take those cells, put them into that person, and they'll develop a new heart. I have not the foggiest notion, nor does anybody else. And we simply don't know what the future has to hold in that respect. What is quite clear, however, is that development is central to our lives. You are only here because of development. And what we hope over the next few years is that we'll be able to offer Frankenstein, and not only Frankenstein, ourselves, a very clear understanding of actually how we come from the egg. Thank you very much for listening.